Hello and welcome to Bio40 Pre-Lecture 9. Today we're going to start discussing DNA. This is the start of our molecular genetics section, which is a personal favorite of mine. The ability to manipulate life on the genetic level is extremely powerful. It makes possible things that used to be reserved for science fiction. The right applications can prevent looming crises the world faces, and the wrong ones can plunge us into a terrible dystopia. The key to all of this lies in our understanding of DNA. This stands for deoxyribonucleic acid, which is a great thing to say if you want to impress people, but otherwise it's best to go with the abbreviation. Let's review some of the history up to this point. In the mid-19th century, Darwin developed a model of evolution, but was missing a good model of inheritance of traits. Unfortunately, he was unaware of his contemporary Mendel, who described laws of inheritance and came up with theoretical particles called genes. Mendel couldn't explain what genes were physically, but a few decades later, Morgan and Bridges were able to demonstrate that genes are in chromosomes. Now this should lead us to ask what chromosomes are, and how do they contain the information that determines traits and allows them to be passed through successive generations. In the early 1900s, the identity of the molecule that contained genetic information in chromosomes was the subject of much debate. Some people suspected it was DNA, while others thought it was protein. A chemical analysis of chromosomes showed both were present in roughly equal amounts. Both are polymers made of repeating monomers, but DNA has only 4 different subunits, while proteins have 20. This means that proteins could contain higher informational complexity. Additionally, DNA has a highly regular repeating structure, while protein structure is much more complex. Most people thought one of these molecules contained genetic information, while the other simply offered structural support to the chromosome. While some people favored proteins due to their higher complexity, by the early decades of the 20th century, more people were beginning to lean towards DNA as the genetic component. Friedrich Miescher had first discovered DNA in the mid-19th century in the nuclei of white blood cells, and showed they were part of chromosomes. And while DNA is primarily found in chromosomes, proteins are distributed throughout the cell. DNA amounts are consistent from cell to cell, while protein amounts vary. Looking at the gametes, there's half as much DNA there as in other cells, which can't be said about protein. Also, when you examine the composition of the molecules, DNA is consistent from cell to cell, while proteins are variable. This pattern for DNA is consistent with somatic cells all having the same number of genes, the genes being identical from cell to cell, and gametes having half the number as somatic cells. As a final note, we should expect the genetic molecule to be very stable to minimize information loss or corruption, and DNA is more stable than proteins. So we have correlations that support DNA as the genetic molecule, but no hard evidence yet. Such evidence did not come about until a series of experiments in the 1920s, 30s, and 40s. Frederick Griffith worked with the bacterium Streptococcus pneumoniae. There are 2 strains of this bacterium that can be distinguished by how they grow on agar plates. One strain forms smooth colonies, while the other forms rough colonies. If you look at them under a microscope, you can see that the smooth, or S strain, appears much larger than the rough, or R strain, due to the presence of a polysaccharide capsule. Only the S strain with the capsule is virulent. When you inject S strain bacteria into mice, the mice die, and if you check their blood, you can find live S bacteria in it. However, when you inject mice with the R strain, the mice survive. The infection is cleared, and there is no streptococcus in their blood. The virulent S strain can be killed with heat, and injection with the heat-killed S strain has no effect. But an odd thing happens when you combine live R strain and heat-killed S strain. Even though neither kills mice alone, the combination is lethal. And not only that, you can find live S strain bacteria in the mouse's blood. So the R strain wasn't just using the capsule from the dead S strain. The actual genetic information about how to form a capsule was transferred to the R strain. At the time, Griffith referred to something in the heat-killed S strain transforming the R strain to S. And we still use the word transformation to describe the introduction of new DNA to bacteria. But at the time, there was no way for Griffith to know that DNA was what was responsible for the change. So the team of Oswald Avery, Colin MacLeod, and Macklin McCarty built upon his work and set out to confirm the identity of the transforming principle. They showed that the mouse host was not necessary for the process. Just mixing heat-killed S and live R strain in a test tube will yield living S bacteria. They then took a biochemical approach and purified different components of the heat-killed S strain to see what was responsible. They found that an extract of DNA, shown here as a white thread-like precipitate, had the transformation ability. But this wasn't enough to convince everyone yet. So Avery, McLeod, and McCarty did another series of experiments we'll discuss in class. Their conclusions were supported by another line of research done by Alfred Hershey and Martha Chase. They studied T2 bacteriophage, a sort of virus that infects the bacterium E. coli. The phage reproduce by injecting their genetic material in the bacteria, and the bacteria does the work of producing more phage. After injecting their genes into the cell, the phage leave behind a ghost, or empty shell, on the cell's surface. Since phage consists of only proteins and DNA, Hershey and Chase only needed to explore these two options as the genetic material. They took phage and grew some in radioactive sulfur and some in radioactive phosphorus. Since sulfur is found in proteins but not DNA, and phosphorus is found in DNA but not proteins, they now had one line of phage with radioactive protein and one with radioactive DNA. They infected separate bacteria populations with each line of phage and looked to see if the radioactivity could be detected in the bacteria and progeny phage, as you'd expect for the genetic material, or outside the bacteria in the supernatant, as you'd expect for the ghosts. They saw that the radioactive protein was mostly in the supernatant, while the radioactive DNA was mostly inside the bacterial cells. And when they looked at the next generation, the progeny of the phage with the labeled DNA had 30 times as much radioactivity as the progeny of the phage with labeled proteins. These experiments might not have been as strong as the Avery, McLeod, McCarty experiments, but they were seen as a confirmation that settled the issue once and for all. With it settled that DNA is a genetic material, a race began to determine the three-dimensional structure of the DNA polymer. And the discovery was first published by James Watson and Francis Crick, building on several key pieces of information discovered by other scientists. First was the knowledge about the individual pieces of DNA. There are three main parts of the DNA monomer: a deoxyribose sugar, a negatively charged phosphate ion, and one of four different nitrogenous bases. Deoxyribose is a pentose sugar, a sugar with a chain of 5 carbons. It has a cyclic form with the 1' and 5' carbons linked via an oxygen atom. Deoxyribose is distinguished from ribose by the groups attached to the 2' carbon. In ribose, the 2' carbon is attached to a hydroxyl group, but in deoxyribose, there is only a hydrogen atom there. While DNA uses the deoxyribose sugar, the related molecule RNA, which we will discuss much more in the future, uses ribose. While every DNA monomer has the same sugar and also the same phosphate, there are 4 different nitrogenous bases each one could have. The bases are flat, or planar, due to all the double bonds present. The bases are separated into 2 classes based on the number of rings. Purines have 2 rings, while pyrimidines have 1. DNA has 2 different purines, adenine and guanine, and 2 different pyrimidines, cytosine and thymine. There is a third pyrimidine, uracil, that is found in RNA in place of thymine. You can see it is very similar to thymine, differing only by the lack of a methyl group on carbon 5. Bases are typically referred to by their first letter. Adenine, guanine, cytosine, and thymine are simply A, G, C, and T. The bases are covalently bound to the sugar's 1' carbon. This is called a nucleoside. The full monomer also has a phosphate attached to the sugar's 5' carbon, and this complete unit is called a nucleotide. The monomers are joined together by phosphodiester bonds. These bonds connect the phosphate of one nucleotide to the 3' carbon of the next nucleotide. This gives a directionality, or polarity, to the polymer. A DNA chain has a 5' and a 3' end. This property will be important for several future applications. So we have the general structure of a single chain here, but that is far from understanding the polymer's 3-dimensional shape. There are 2 more big discoveries necessary to get to that point. The first was by Erwin Chargaff. Chargaff took DNA from several different species and hydrolyzed it, or broke the polymer down to the individual nucleotides. Then he used paper chromatography to separate the bases and quantify how much of each was present. His results are in this table, and based on these results, he came up with some general rules. The amount of adenine equals the amount of thymine. and the amount of guanine equals the amount of cytosine. However, the amount of adenine and thymine together didn't necessarily equal the amount of guanine and cytosine. Further, the ratio of A's and T's to G's and C's weren't consistent across species. In some species they were close to equal, but some had many more A's and T's, while others had more G's and C's. This was certainly interesting, but the importance was not immediately clear. The other big piece of the puzzle was X-ray crystallography data collected by Rosalind Franklin. In X-ray crystallography, you first get the molecule you're studying to form crystals. Then you shoot X-rays at the crystals and observe how the molecule's electrons cause the X-rays to change course or diffract. Diffraction patterns like the one shown at the bottom right consist of tons of tiny dots, each the result of an X-ray diffracting and striking a piece of film. The angles of diffraction can tell you the shape of a molecule's electron cloud, and thus the molecule itself. These days, powerful computers do the work of interpreting these patterns, but in Franklin's time this was all done by hand. Many people tried using X-ray crystallography to get the structure of DNA, but Franklin was the best crystallographer working on this. She was able to crystallize multiple forms of DNA called A and B. She determined the major form, the B form, had a helical shape, likely 2 helices. She saw the molecule had periodicity, or repeating patterns. One pattern repeated every 3.4 angstroms, while another repeated every 34 angstroms. Franklin also saw the molecule had a 20 angstrom diameter. For reference, an angstrom is 10 to the negative 10 meters, or 0.1 nanometers. This was the information necessary to deduce the structure of DNA. Next time, we'll discuss that structure and some of its implications.